Sterile pyuria. Pyuria is defined as the presence of 10 or more white cells per cubic millimeter in a urine specimen, 3 or more white cells per high-power field of unspun urine. A positive result on Gram's staining of an unspun urine specimen, or a urinary dipstick test that is positive for leukocyte esterase. Sterile pyuria is the persistent finding of white cells in the urine in the absence of bacteria, as determined by means of aerobic laboratory techniques on a 5% cheap blood agar plate and McConkey agar plate. Sterile pyuria is a high prevalent condition, and population-based studies show that 13.9% of women and 2.6% of men are affected. Specific populations have a higher risk of this condition. For example, the frequency of detection of sterile pyuria was 23% among inpatients in one study, excluding those with urinary tract infection. And sterile pyuria is more common among women than among men because of pelvic infection. Causes of sterile pyuria: sexually transmitted infections, gonorrhea and chlamydia, genital herpes and herpes zoster, HPV and human immunodeficiency virus infections. Other viral infections such as adenovirus, BK poliovirus, and cytomegalovirus may cause hemorrhagic cystitis in immunocompromised children. However, these infections are typically not associated with pyuria. Genitourinary tuberculosis. Genitourinary tuberculosis, the most common form of non-pulmonary tuberculosis after lymphadenopathy. Hematuria and pyuria are typically findings in genitourinary tuberculosis. This condition can infect the kidneys, ureters, bladder, prostate, and genitalia. Genitourinary tuberculosis can cause renal calyceal destruction, calyceal obstruction, or hydronephrosis, or all these conditions. Since the incidence of tubercular infection is 13 to 26 times as high among foreign-born persons and recent immigrants as among non-Hispanic whites, clinical suspicion of tuberculosis infection should be higher in these patients when they present with sterile pyuria. The tuberculin skin test is helpful in determining whether a person has been exposed to tuberculosis, but false positive results often occur in patients who have received the Mycobacterium curie (BCG) vaccine, and a false negative skin test may occur in patients with impaired T cell function. Interferon Y release assays are whole blood tests that are not affected by BCG immunization. Fungal infections. Candida infections are a common source of urosepsis in hospitalized patients. especially those who are immunocompromised. Candida albicans is the most prevalent species. However, C. glabrata, C. tropicalis, C. crusei, and other Candida species can also cause infection. Notably, patients with diabetes are prone to Candida infections. Patients who have received transplants are vulnerable to aspergillosis, and patients with HIV infection may be susceptible to cryptococcuria, blastomycosis, coccidioidomycosis, and histoplasmosis are associated with intensive environmental exposures. Example, disruption of the environment by construction, sandstorms, or tornadoes, or exposure to a high concentration of bird excrement. All these fungal infections may cause genitourinary infection with associated pyorrhea. Urine microscopy may show budding yeast from or hyphae, but identification of fungus requires a special culture medium and from 3 days to 3 weeks for speciation. 32 in patients with Candida or Aspergillus infections. Imaging studies may reveal filling defects in the collecting system or bladder caused by fungal materials that are referred to as fungal balls. Evaluation of patients with steroid pyuria. The differential diagnosis of steroid pyuria is broad. A complete history and physical examination with consideration of the factors listed in are required to identify the potential causes of genitourinary inflammation. A specific evaluation for sexually transmitted infections is warranted. Evaluation to detect bacterial, fungal, and parasitic infections is indicated in patients with a clinical history that suggests specific infections. Abdominal, renal, and bladder imaging shall be considered for evaluation or febrile or otherwise symptomatic patients. Inflammatory conditions near the urinary tract, as well as systematic diseases, should be included in the differential diagnosis. Steroid pyuria has historically been considered to be suggestive of genitourinary tuberculosis, but a wide variety of these causes must be considered. Criteria for successful treatment of conditions that cause steroid pyuria include curtailment or resolution of symptoms, a negative culture, or a negative PCR assay. Pyuria may persist because of underlying inflammatory changes.